in writing. Now to introduce today's speaker, Mason Kamalani Chock, who goes by Kama. He is a PhD student researching microbial community ecology at the University of California, Berkeley. Kama, I'm just going to pass the screen to you.
Perfect.
Okay, you should now be able to accept the invitation to present. Can you see it there?
I do.
There we go. A native Hawaiian, Kama did his master's in botany at the University of Hawaii, and he will be presenting work from that time today. Rightio, Kama, I think you should be ready to begin your presentation.
All right, yeah. Well, thanks so much, Renee. Yeah, this is an awesome webinar series, so thank you for having me on it. I feel very special to be on it. So today I'm going to be talking about some work I did during my master's at the University of Hawaii, Manoa. And I was working in Anthony A. Mann's laboratory, and we kind of focused on looking at the fungal communities that live inside of leaves and seeing how we can harness them to increase disease resistance, specifically in plants that are threatened by introduced disease, such as Myrtle Rust. So let's see. All right. So just a slide on some background about Myrtle Rust in Hawaii for those who are unfamiliar. So Myrtle Rust was introduced in 2005 onto the island of Oahu. And within 8 months, I think it quickly spread throughout the entire island chain. But luckily, I think similarly to a lot of other countries in the Pacific, there's only a single known genotype that is currently inhabiting the state, which is the pandemic biotype. But there were some studies in 2014 looking at the virulence of other strains present in Brazil on Ohi'alehua, which is the dominant canopy of Hawaiian rainforest, and they do seem to be fairly virulent. So there are some dangers of those strains. And in addition, there are also some dangers of changes in climate due to anthropogenic causes. So here's just like a max ent, just a basic max ent model map of Hawaii, and seeing how the spread, the range of Myrtle Rust might change over time with changes in temperature. So yeah, I mean, it's definitely a big problem in Hawaii. Besides Ohi'alehua, Myrtle Rust affects a few other species. I think actually it infects all native, 7 native species of Myrtaceae in Hawaii. So there are 5 species of Ohi'alehua, Metrosideros spa. Most people are probably familiar with Metrosideros polymorpha, which is probably the most widely distributed across the island. But there are 2 other species of Eugenia. We call them nioi in Hawaii. And one of them is kind of this commonly found Eugenia renwardiana. And there's also the critically endangered Eugenia koolauensis, and that's what we're going to be talking about today. And as is the case for a lot of endangered plants, unfortunately, Eugenia koolauensis is the most susceptible to Myrtle Rust. But yeah, and all of these kind of have these pretty typical Myrtaceae flowers with the clusters of stamen. But yeah, so a little more background on Eugenia koolauensis. I'll just call it nioi from now on. It's a bit of a mouthful otherwise. But yeah, like I said, it's critically endangered. Nowadays, it's restricted to about 99 trees distributed across 11 populations on the island of Oahu. Historically, I believe it was on Molokai and maybe another island. But yeah, it's very sparse nowadays. But luckily, there is an organization, Oahu Army Natural Resource Program, headed by Kapua Covello, and a lot of great work done by Matt Garma as well in the greenhouse, where they're actually creating this huge stockpile of germplasm to kind of save these plants from extinction, at least ex situ. So they have about 300 genotypes, I believe, of Neoi in triplicate in their greenhouses. Unfortunately, it requires these populations require a regular application of fungicide, I think like every 2 weeks, because of how prevalent the Myrtle Rust is even in the greenhouses. But yeah, here's just some pictures of Neoi. It's a really big tree. Starts off as really small seedlings, and it kind of has this characteristic cute little orange fruit. So yeah, so besides, I guess, right, so sorry, before I get ahead of myself, even though they're doing a lot of great work with its ex situ populations, even though when they try to outplant these plants into the wild, they kind of immediately succumb to the rust just because of how virulent it is on this species. And so the regular application of fungicides is good for controlling the rust, but it has a secondary effect, which is unfortunate. So from the work that we do in the AMAND laboratory, we know that there are within leaves there are thousands of fungal species that exist, and a lot of them are beneficial. So by applying fungicides to these leaves, you actually create a depleted microbiome. So you're getting rid of all of these potentially beneficial fungi that exist, and this allows basically allows free real estate for this Myrtle Rust to recolonize and eventually cause disease. Right. And so, right, and you can kind of make an analogy towards, I think it's really helpful to make an analogy towards the gut microbiome in humans. So when we take, this is a good parallel where, for example, if we were to take antibiotics, it would kind of deplete our microbiome in our gut, and it causes some kind of dysbiosis if there are other non-commensal or even pathogenic bacteria or fungi that are introduced to our guts. So our idea in this project was, after being depleted by fungicides, was to reintroduce these beneficial foliar endophytic fungi, and you can call them FEF. I'll probably call them that for the rest of the presentation. So by introducing these beneficial FEF back into the leaf community, we can kind of get rid of that free real estate, take it up by these beneficials, and prevent the rest from recolonizing the leaf, and hopefully create or maintain a healthy leaf microbiome and a healthy leaf in general. So a little background on FEF. It's pretty well known. I mean, these have been studied since the 1900s. And so there are a bunch of different benefits that they provide for plants, including just general plant growth, increased stress tolerance, and what we're interested in is increased pathogen resistance. And it's nice to work with these because FEF are found pretty much in all living tissue. So if you were to go to your backyard and pick a leaf from one of your plants, you'll most likely find a lot of these guys living in there. So yeah, they provide a really nice potential disease biocontrol agent, especially for Myrtle Rust. Right, and so there are a bunch of different mechanisms that FEF are able to use in order to inhibit pathogens. So there's direct mechanisms where they can release secondary metabolites, or just simply competitively exclude the pathogens. So they're basically just taking up the real estate of the leaf. And then there are a little more complicated ways of pathogen inhibition, which is host manipulation through some like genetic cascades or something like that. And then also these FEF don't just exist by themselves. They can also recruit other microbes that are beneficial that help out with inhibiting the pathogen. Right, so the first step that we did in this experiment was to isolate as many FEF from both healthy and diseased leaves of related species that are susceptible to Myrtle Rust. And so I probably had like a few hundred of these plates, but we separated them by morphotype, and in the end, we identified 46 distinct foliar fungal taxa. Yeah, and so once we had this library of fungal taxa, we kind of created this standard experimental design that included the propagation of the plants. So OANRP was really helpful for helping us start these plants and giving us a lot of good tips on how to grow them. And so once their microbiome was depleted by fungicide, we wanted to reintroduce microbes to their leaves. So we inoculated them with foliar or FEF, waited 72 hours to help them establish within the leaf community, and then we inoculated the rest onto the same leaves. And we kind of just used the standard procedure where with these little humidity bags in darkness for 12 hours. Right, and then we just did some analysis. We used this Weka segmentation analysis in ImageJ. It's just a nice package for quantifying rust on leaves. And so yeah, this is the general experimental design. But before we actually implemented this, we wanted to choose which fungi were actually might be inhibitive to Myrtle Rust. So we did some scanning electron microscopy. Ben Hoyt, who is now a PhD student at UC Riverside, he was an undergrad at UH Manoa before. We did some scanning electron microscopy of specifically seeds that were, not seeds, leaves that were inoculated with rust. And so we saw some interesting interactions. This is an example where we saw a bunch of fungal growth within the actual pustules. And so we isolated that fungi, and then we also saw some interesting interactions with hyphy that were wrapping around some spores of Myrtle Rust. So we kind of were interested in that as well. So we isolated a bunch of taxa from those leaves and came up with a few candidate FEF to use in these treatments. So we had these technically 3 taxa, Cladosporium, Bionectria, and Clonostachys. And for the Clonostachys rosea, it was particularly prevalent in those hyperparasitic leaves. So we were just kind of interested in that in particular. So we had 4 treatments of spore slurries of these fungal taxa. And then in addition, this was actually kind of a last minute addition, but we decided to include a leaf homogenate treatment as well. So with the thought of introducing not just a single taxa, but a whole community of FEF. With the thought that with more taxa, we might create some kind of portfolio effect where we would have just a higher chance of encountering a beneficial FEF. So for this treatment, we basically just blended up leaves, pulled it through a 100 micrometer filter, and then sprayed that onto the plants. And so, right, this is a kind of a representation of a whole intact community of FEF. Right, and so just as a summary, we have 4 fungal spore slurry treatments, a leaf homogenate treatment, and a few controls, so negative, positive, and then also a control for the leaf homogenate. And right, so there's a lot going on in this figure, but let me just break it down a bit. So in this first box, we found that kind of what we expected. So the rust only treatment, which is just rust sprayed onto the leaves, gave us a kind of a threshold for disease severity. And then our negative control had no rust, unsurprisingly. And then our leaf homogenate control also had an insignificant difference between the positive control. And so with the leaf homogenate treatment, this middle column here, we did see what we thought, this wasn't actually statistically analyzed yet, but what we thought was a statistically significant effect of the leaf homogenate treatment. And then the four spore slurries, right, of the different taxa. Yeah, and so after looking at the not just the p-values, but also the effect sizes of each treatment, we could see that the negative control was what we expected to be lower than this middle line, which is the threshold for the positive control. But we are also surprisingly saw that the leaf homogenate treatment was the only treatment to actually decrease Myrtle Rust growth in this experiment. And so this experiment kind of just showed that these single species inoculum are not particularly effective at inhibiting rust growth, and that we should probably be considering, at least in these techniques, to be using a whole community inoculum for inhibiting Myrtle Rust. Yeah, I think that went a lot faster than I thought it was going to be, but hopefully it was interesting. I'd like to thank everyone that helped me out with this project during my master's, my committee, Anthony, Casey, and Nicole, Tina, who helped me out with the scanning electron microscopy, Janice Uchida and Chris Kaduka, who have done a lot of research in Hawaii with Myrtle Rust, and then the rest of the lab, which were very helpful. Yeah, and I also want to give a big shout out to Oahu Army Natural Resource Program, who helped a lot in this project. Yeah, and if you're interested in making cool figures like I had today, check out BioRender as well. Anyway, yeah, I just want to thank you guys so much for having me. Yeah, and I'm happy to answer any questions.
Thanks, Kama. Thanks for that excellent talk. We do have the first question. I'm not sure if I've quite shared it successfully with the audience, but I'll ask it here.
Sure.
If the neoi is highly susceptible, why do you think the foliar microbiome would inhibit the rust? Perhaps the thought is these plants must already have some kind of foliar microbiome and it's not working.
Right, so they're
back in touch the person if asking that question if I haven't quite interpreted your question correctly.
Yeah, I mean, I can try to elaborate on that. So by being constantly bombarded by fungicides, right? So this kind of causes a type of dysbiosis in the foliar microbiome. And so by reintroducing an intact community, maybe it's restoring a lot of functions to the actual plant that maybe are not just related to the Myrtle Rust resistance, but also just general plant function as well. Yeah, I mean, it's hard because this was almost like a preliminary study, right? We didn't actually verify what the mechanisms behind the inocula were for inhibition. So yeah, I mean, there's a lot of speculation, and I can speculate with the audience. I mean, I have a few other slides we can talk about as well.
Well, for sure. I mean, yours is one of these classic studies that asks as many questions as it answers, but of course this is not unusual at all. Okay, well, Yeah, this is a million-dollar question, of course, for a lot of this work. Are you aware that at least some of the fungal members of the community won't be culturable? What
Yeah.
should we do about them? How are we going to consider them?
Yeah, no, that's a great point, especially so right in the early 2010s, right, this explosion of microbiome papers became a thing because of this technology of high throughput sequencing, and it gives us a better idea of what's present in the microbiome, but something like over 95% of the microbes that we find in those studies aren't culturable. So what we tried to do for our study was re-isolate a bunch of, whatever was growing inside of that inoculum, that leaf homogenate inocula. And yeah, I mean, those could be used for further studies, but we don't actually know the culprit behind the disease resistance. There was so another study in the lab did something very similar, and they basically all you have to do is correlate pathogen infection load with whatever taxa are abundant at the low infection loads, and then if you are able to isolate those, then you can use those in follow-up studies to test if they are actually inhibiting the disease. So, yeah.
Right. Did you consider the effect of the foliar bacteria?
Yeah, so that was kind of the reason for and I didn't fully explain it, so apologies for that, but that was the reason for this leaf homogenate control. So we've pulled the leaf homogenate further through a 2 micrometer filter for these purple dots treatment, and with the thought that by filtering out the fungi, that we are kind of I don't know, canceling out the idea that bacteria are part of that pathogen inhibition. But there was, I mean, it wasn't significant, but I mean, there was like a slight decrease in pathogen inhibition. So it's very possible. And I don't think you can rule it out completely. That's a great point.
For sure, and we're getting plenty more questions coming through here, and I guess an important thing for people to remember is that this is just a master's. I shouldn't say just and diminish your efforts, but of course, one is limited in what one can do.
I
When
appreciate
I
that. No,
presume you did this in 1 year.
yeah,
Uh,
about, yeah.
yeah, and largely independently. Right. So you probably didn't have a chance to look into what might be in the Jambos leaf homogenate that might really be driving this effect, oils, endophytes, etc.
Yeah, I mean, so we just have the re-isolated, whatever we could re-isolate from that homogenate. But we did not, I mean, it would be easy to, now that I'm doing this amplicon sequencing every day, it's quite easy to do now, I realize, but yeah, it would be totally possible to go back and isolate, I mean, not isolate, but sequence that homogenate to see the general composition of that treatment.
And I guess in a similar vein, from grass endophyte experiences, we know secondary metabolites play a crucial role.
Mhm.
Did you look into, or will anyone look into the fungal secondary metabolites present?
Yeah, no, unfortunately. Yeah, I mean, it's just crazy how many different types of mechanisms that might be behind this and
For
this
sure.
pathogen. But yeah, that was another great one. Um, yeah, yeah.
This is a good one. Have you evaluated applications of these endophytes without the application of fungicide?
No, actually. So well, it would be difficult with this particular species because um,
The rust would probably get in very fast by the sounds of it. It's
yeah, I mean,
quite amazing.
in a greenhouse, it's in an enclosed greenhouse, it is definitely possible if you keep them away, and especially in a growth chamber, actually. So, but yeah, I don't think it would be very representative of the actual natural microbiome though. It would be like this kind of, whatever would be on the leaf after or before fungicide application would be whatever is growing in that growth chamber, which might not be, yeah, completely representative of a natural ecosystem, but I mean, it would be interesting. I definitely agree.
Did you, or how did you confirm colonization by the foliar endophytes in the transplants?
Could you say that one more time?
Did you confirm colonization by the endophyte? So did you take a look to see what had actually established, and if so, how did you do that?
Oh yeah, yeah. So we did plate some of the leaves that we didn't analyze, and I didn't see growth of, I couldn't re-isolate this cladosporium, but we did re-isolate these other 3 taxa from the leaves.
Okay, very good. How did you determine whether all the foliar endophytes were eliminated prior to re-inoculation?
Yeah, that's a good point. Actually, we didn't control for that. So no, yeah, I mean, that was definitely an oversight, but yeah.
Okay, and I'm struggling a little with this one. Is the plant with the intact community highly susceptible or not? Not quite sure what the questioner means by intact
Oh
community.
yes. So is Neoi just regularly, um, yeah, maybe I don't fully understand it, but is Neoi regularly susceptible? And it was not, it's not quite as susceptible as Syzygium jambos, but it is pretty regularly susceptible.
Yes. I guess this is one of the challenges of this webinar platform where I need to ask the questions, and perhaps I'm not always going to get it right.
Well, I'm understanding it at least for the most part. So
All right, now we have results were not significant. It's not clear, seems they're not. Well, they are, right, for this intact community. It being,
for the leaf homogenate, yes, it was significant.
yes,
Um,
right. For the homogenate that showed some efficacy, is the next step to fractionate this and identify the microorganisms that were primarily responsible.
yes.
And I guess this kind of links into another question: what is the next step? Will it be repeated? I mean, you've moved on to new horizons and a PhD opportunity. So are you aware of what's going on back there in Hawaii if others are taking this work further?
Yeah, no, so I mean, that's one of the downsides of academia where it's kind of, it's a lot of transientness. But
Yeah, absolutely.
so the Amen laboratory is they're primarily interested in fungal biogeography, and they're actually I was kind of in a transition period where they started to look at a lot of marine fungi. So I don't believe that anyone's picked up the project, but also the funding for the project. I kind of got the tail end of this other grant to do this project, so I am not too sure. Yeah, but
Well,
honestly, if any PhD students or prospective PhD students are interested, please contact Anthony. He's great to work with, and yeah.
well, for sure. And we do have, I can confirm, some experts in microbiome and foliar endophyte work present in the webinar today. So I'm sure they'll take something from what you've done and have really enjoyed your presentation. All right, another question here, although we will be wrapping it up very shortly. Was the S. jambos leaf collected from a natural population that was exposed to a psidii?
Yes, it was. So there actually probably was some Myrtle Rust in the actual leaf homogenate. So I would be surprised if there wasn't.
And more of a comment here, I guess you should consider to get organisms from plants that show some resistance. Well, yeah, I'm sure others will be interested in doing that, thinking about doing that.
Yeah, no, that's a good point. And I think this project in general, I think the main thing we can take away as a community that's interested in kind of fighting back this Myrtle Rust in general is if we are interested in looking at microbial applications for disease resistance against Myrtle Rust, I think it's going to take not just a single taxa, but multiple, or at least the whole community of microbes to fight off this.
Yeah, I mean, the endless debate about the portfolio effect rages on, though, doesn't it? Really, is it just that one magic thing, or is it about things interacting with each other in the plants?
Yeah, and
You know,
I think that's
one
why I've
study,
gravitated. Oh, sorry.
one study will say one thing, and another will suggest another. At this point.
Exactly. So I think that's why I've kind of gravitated towards the research what I'm doing right now, which is looking like really breaking down, looking at stuff from a bottom up perspective rather than a top down, which this experiment kind of was, and looking at the actual mechanisms that are occurring or that are causing differences in disease resistance in relation to the microbiome. So there's a lot of work to be done, but yeah, no, it's definitely exciting, and yeah, I think it's
For sure,
definitely.
and I guess just one final, perhaps self-indulgent question from me. I was just curious about this neoi tree. They're obviously quite ginormous and very, very tall. Has anybody ever tried to get right up into that top canopy and check what's going on with the new foliage up in these very mature specimens?
Yeah, no, that's a great question. I'm not too sure. Yeah, it would be interesting to see what's happening with the healthy leaves and the healthy plants of neoi in the wild. I think that, yeah, that would totally be something worth looking at.
For sure. I mean, I have met some expert tree climbing ecologists in my time who I'm sure would be delighted to have the chance to get up
Yeah,
one of those and see what's going on
absolutely.
up the top. All right, well, that's it. Thank you very much. A video of today's webinar and answers to questions will be made available on the Beyond Myrtle Rust website in the next couple of days. And now to bring up the next webinar, that will be Wednesday, July 14th, when we will hear from Phil Cannon, a forest pathologist with the US Forest Service, currently based in California. Now, Phil has a long history with New Zealand, as well as with Myrtle Rust. In fact, Phil has previously lived and researched here, and he visited here to advise on New Zealand's response when a psidii first turned up. We will again be briefly visiting Hawaii, with his talk being titled Experiences with Austropuccinia psidii in Hawaii and the Americas of Relevance to the Land Down Under. Thanks again, everyone. It was great to have you along, and we'll see you next month.